I am going to explain the T cell tolerance. Tolerance means unresponsiveness to self tissues, to self antigens, to self organs. So T cells originate from bone marrow. However, when they originate from bone marrow, they are CD3 positive and CD8 negative. This is precursor of T cytotoxic, and CD4 negative, and this is a precursor of T helper cell. And the TCR, it is only beta and pre-alpha, premature. So when they enter the thymus gland, why T lymphocytes migrate from bone marrow to thymus gland? They migrate from bone marrow to thymus gland for maturation, for development of a repertoire. Development of a repertoire means that they express unique receptors for different antigens or pathogens in the environment even before they encounter that environment. Okay? Third, they get an education. How do they get education? Thymus gland, it looks like a school. So T lymphocytes, they come here, T lymphocytes, they come to thymus to get education. As the cortex, this is gut section. Okay, these are trabeculae. This is the capsule of the thymus gland. So immature T cells, they enter through high endothelial venules as immature and interact with the thymic epithelial cells. These thymic epithelial cells express major histocompatibility class 1, major histocompatibility class 2. So if these immature cells reacted with major histoclass 2, they will get differentiated into CD4. If they interacted with major histocompatibility class 1, they will get differentiated into T cytotoxic or CD4. And this is here, at this stage, they should recognize the major histocompatibility class 1 and 2 as self. And this is known as positive selection, and ensures major histocompatibility restriction. So they should in the future time interfere with non-self or with pathogen in association with major histocompatibility. However, now this is the first lesson, they got it, and that failed to get this lesson, or that thymocytes failed to recognize this major histocompatibility molecules, they will be killed by apoptosis. And so here, but however, the positively selected, this we call it positive, they go deep, okay, and then they interact with macrophage and dendritic cell, and now they can see the body antigens. So these cells here in the thymus gland, dendritic cells and macrophage, they present body antigens, antigens of the heart, antigen of the muscle, antigen of bone, any antigen in your body except two antigens, they don't present it. Antigen is sperms and cornea. So the immune system should not see sperms, should not see the cornea. But however, these cells now, they can see antigens from all tissue of the body. And here, if they recognize the self-antigen as self, and if they recognize it, the self-antigen in association also with major histocompatibility, self-major histocompatibility, they will be saved. But if they fail to recognize the self-antigens, they will be killed by apoptosis, and this is known as negative selective selection. Negative selection for insurance of self tolerance. So only 5% of T lymphocytes exit the thymus gland, and around 95% of thymocytes arrive to thymus will be killed by apoptosis. So, however, in some instances, some autoreactive T cells may escape, and some autoreactive T cells they express some cluster of differentiation. It is known as cluster of differentiation number 25, and they become a regulatory T cells. So negative and positive selection ensures self-tolerance, tolerance to self-antigens encounter in the thymus is thereby achieved by elimination or eliminating T cells that are reactive to self-antigen. So positive and negative selection of thymocytes result in mature T cells, as can be seen here, are both self MHC restricted and self tolerant. And as you know that SLE is hypersensitive reaction type 3, because immune complexes in case of systemic lupus erythematosus are circulating immune complexes, and they are not membrane bound immune complexes. Thank you very much, Professor Ahmed Bolad.